Swayam Prabha. Digital India. Educated India.
We'll continue our discussion about the regulation of gene expression by G protein coupled receptors. This is what we have been discussing in the last 3 classes. How signaling molecules, when they interact with specific receptors, specifically the serpentine receptors or the 7 transmembrane domain receptors, leading to the results in the activation of trimeric G proteins, and this results in the dissociation of the beta, gamma and alpha subunits, and they then interact with specific effector molecules like either adenylate cyclase or phospholipase C, resulting in the second generation of specific second messengers, and these second messengers ultimately go on then phosphorylate, activate specific kinases, resulting in the activation or repression of specific genes, as well as activation or repression of metabolic enzymes, ion channels and so on and so forth. What we have discussed in the last 3 classes is how signaling molecules interact with G protein coupled receptors. This results in the activation of trimeric G proteins, and we discussed primarily about those G proteins which ultimately activate protein kinase A signaling, protein kinase G signaling, and in the last class we discussed briefly about the generation of second messengers such as inositol triphosphate, diacylglycerol and calcium, and how these 3 molecules, when they are generated by specific signaling molecules, results in the activation of protein kinase C as well as calcium calmodulin kinase and so on and so forth. So what we will do today is we are going to spend considerable amount of time to discuss about protein kinase C, because it is a very important protein kinase, and we will discuss about how protein kinase is generated by varying signaling molecules through the G protein coupled receptor pathway, and what kind of we will discuss some examples of genes or gene expression pathways which are activated by protein kinase C. So we have been discussing primarily about membrane receptors and how ligands or hormones, when they bind to these membrane receptors, lead to the activation of specific signaling pathways, ultimately culminating in activation or repression of genes. So far we have discussed about receptors that regulate cyclic AMP formation, receptors that regulate cyclic GMP formation. Today we are going to spend some time about the receptors that regulate diacylglycerol and inositol triphosphate formation, and how these molecules in turn regulate gene expression, and maybe in the next class we are going to discuss about receptors that possess protein kinase activity. So since we have already covered the first 2, today we are going to finally focus on the third kind of membrane receptors, namely those receptors which activate G proteins that ultimately results in the activation of phospholipase C, resulting in the generation of diacylglycerol and inositol triphosphate, and how these act as signaling molecules. As I just mentioned, of the various trimeric G proteins that we have been discussing, so far we have discussed about the GS alpha subunit, which primarily activates adenylate cyclase, leading to an increase in cyclic AMP. There are also molecules which activate the GI G alpha I subunit, resulting in the downregulation of cyclic AMP by actually activating phosphodiesterases, and we also then discussed about the cyclic GMP pathway, where the membrane receptor itself contains intrinsic cyclic adenylate guanylate cyclase activity. This results in the synthesis of cyclic GMP, and then how cyclic GMP regulates gene expression, especially in a calcium dependent manner. Today we are primarily going to focus about trimeric G proteins contain the G alpha Q subunit, and when they are activated, how they activate phospholipase C, so that it results in generation of inositol triphosphate and diacylglycerol. We have already discussed in the last class quite a bit about this particular aspect, but we will just briefly go through what we have discussed and then get into the mechanism by which protein kinase C acts as a important activator of various gene expression pathways. As I said, when we talk about the membrane receptors, especially the G protein coupled receptors, there are 3 major enzymes that we have to remember. One is the adenylate cyclase, which we have already discussed, which basically converts ATP to cyclic AMP and pyrophosphate, and the cyclic AMP also is converted back by phosphodiesterase to ATP, and this results in the reversal of signaling. Regulation takes place both by cyclic AMP generation as well as by cyclic AMP cleavage by phosphodiesterases. We also discussed about guanylate cyclase, which is again a very important signaling pathway, how GTP is converted to cGMP, and then again how cGMP converted back to GTP phosphodiesterases, and how these signaling events ultimately results in activation or depression of transcription. So today we are going to focus about the third important enzyme in the GPCR signaling pathway, namely the phospholipase C or the PLC, which basically converts phosphatidylinositol 4,5 bisphosphate or PIP2 to inositol triphosphate or IP3 and diacylglycerol or DAG. So the focus today is going to be about this particular aspect. So signal transduction through receptor triggered hydrolysis of phosphatidylinositol 4,5 bisphosphate primarily happens when a receptor which is activated by a signaling molecule, it could be a ligand, it could be a small molecule, or it could be a polypeptide hormone, that results in the stimulation of GTP to GDP exchange on a trimeric G protein, and those G proteins which activate phospholipase C, the alpha subunit of are referred to as GQ and alpha subunit is referred to as the G alpha Q subunit, and the GQ actually activates phospholipase C. So when this activation takes place, the membrane bound phospholipase C hydrolyzes the phosphodiester bond linking the phosphatidylinositol unit to the acylated glycerol moiety, and as a result of this hydrolysis, it results in the generation of two very important intracellular signaling molecules, namely IP3 and DAG, that is inositol triphosphate and diacylglycerol. This is a schematic which basically explains what we are going to discuss today and what I have been discussing so far. Basically, when the G protein gets activated, the G protein basically activates the phospholipase C, and when the phospholipase C is activated, the PIP2 that is phosphatidylinositol bisphosphate is converted to diacylglycerol and inositol triphosphate, and the inositol triphosphate then interact with specific receptors on the endoplasmic reticulum, and as a result the calcium which is present inside endoplasmic reticulum is now released, and the calcium thus released now along with diacylglycerol activates protein kinase C, and this activated protein kinase C now goes and phosphorylates a number of target proteins which could be metabolic enzymes, ion channel proteins, or transcription factors. But as I have been telling all the time, these second messenger molecules, whether it can be cyclic AMP or cyclic GMP, calcium, or IP3 or diacylglycerol, they perform pleiotropic functions, but in this lecture series we are going to focus primarily on how these second messengers ultimately activate the regulation of gene expression. So we are not going to talk too much about the activation or repression of metabolic enzymes or activation or repression of ion channels by these second messenger molecules, although they are very, very important from the biology and medical point of view. So this is the structure of the phosphatidylinositol 4,5 bisphosphate, and when this PIP2 is cleaved by phospholipase C, it basically results in the generation of inositol triphosphate and diacylglycerol as is shown here, which are the two signaling molecules, and how they are going to act is what we are going to discuss today. So phospholipase C actually cleaves this particular phosphodiester bond. As a result, hydrolysis of this bond results in generation of diacylglycerol and inositol triphosphate. You have inositol 1, 2, 3 triphosphate, and this is the diacylglycerol, where you have the glycerol backbone linked to two fatty acid chains, so diacylglycerol. Now, phospholipase C is a cytosolic enzyme. It acts on membrane inserted phosphosinositide substrates. There are actually two types of phospholipases. One is called phospholipase C beta, which is activated by G protein coupled receptors. This is what we are going to discuss today. There is also another phospholipase C which is called phospholipase C gamma, which is primarily activated by receptor tyrosine kinases, which we are going to discuss maybe in the next or the coming classes, in the next few classes. So this is basically the various structural domains of the phospholipase C beta, which contains various domains. As far as the regulatory point of view, it is the C2 domain which actually interacts with the G alpha subunit of the GTPase, and the pH domain actually binds to the PIP2, phosphatidylinositol bisphosphate. Both diacylglycerol and inositol triphosphate, which have been produced by the action of phospholipase C on phosphatidylinositol bisphosphate, are very, very important second messengers, and they act on a number of they perform number of physiological functions. These are first discovered in insect salivary glands in the early 1980s, and some of the important functions include activation of platelets, muscle contraction, antibody secretion, and so on and so forth, in addition to activation of gene expression programs. The diacylglycerol is a very nonpolar molecule in contrast to IP3, which is a hydrophilic molecule. The DAG therefore remains membrane bound. You can see The red actually indicates the polar groups. It is largely hydrophobic and therefore it remains associated with the hydrophobic membranes, whereas the IP3 contains a lot of charged surfaces, number of polar groups, and therefore it remains soluble in the cytoplasm and it can easily diffuse through the cytosol. So, the inositol triphosphate, which has been synthesized by the action of phospholipase C on phosphatidylinositol bisphosphate, rapidly diffuses through the cytoplasm, and its primary function is to rapidly release calcium from intracellular stores, calcium stores like the endoplasmic reticulum, or if it is smooth muscle cells, it is called the sarcoplasmic reticulum. So, the endoplasmic reticulum, which acts as the storage of calcium, the calcium is released from these storehouses by the action of inositol triphosphate. On the other hand, the diacylglycerol, which is also the other molecule which has been synthesized which is made by the cleavage of phosphatidylinositol bisphosphate by phospholipase C, remains in the membrane. It has two very important roles. One is it can be cleaved, it can be further converted to arachidonic acid, or it directly goes and activates protein kinase C along with calcium. So, our talk is primarily going to focus upon how DAG activates protein kinase C and what does protein kinase C activate in the further downstream signaling pathways. But I am going to just spend 1 minute just to introduce to you about the importance of arachidonic acid, which is another very important effector molecule that is synthesized from diacylglycerol. So, do not be under the impression that diacylglycerol primarily is involved in the activation of protein kinase C. Diacylglycerol is also converted to arachidonic acid, and as you can see, arachidonic acid is a very important molecule and is the precursor for the synthesis of a number of molecules called eicosanoids. Now, what are these eicosanoids? Eicosanoids are the molecules which are actually involved in a number of physiological processes, and the first enzyme involved in their synthesis, namely cyclooxygenase, is actually the target of aspirin. I am sure most of you have taken aspirin tablets whenever you have pain, or when you are a heart patient, when you want to actually keep your blood thin. People usually recommend you take aspirin, and you can see the two important properties of aspirin is that it reduces platelet aggregation and therefore prevents blood clotting, and it reduces inflammation and pain, and it does both this process by actually inhibiting molecules called the enzyme called cyclooxygenase. And cyclooxygenase is the first enzyme which converts arachidonic acid into the eicosanoids. So, diacylglycerol is not only involved in the activation of protein kinase C, but it also is the precursor for the synthesis of eicosanoids, which play a very important role in inflammation, pain, as well as platelet aggregation and blood clotting. So, just remember this, but we are not going to discuss this point in detail, but it is a very important point to remember, because this particular pathway involving the synthesis of eicosanoids as well as the enzymes of this particular pathway is a billion-dollar industry, and number of molecules that we are using today are all involved either activators or inhibitors of this particular enzymes involved in this particular pathway. Now, let us come back to protein kinase C. So, once the diacylglycerol is synthesized by the action of phospholipase C, protein kinase C now has to go and then phosphorylate a number of effector molecules. Let us now discuss very briefly about what is protein kinase C and how was it discovered. Protein kinase C was actually discovered in the year 1977 in the laboratory of Nishizuka in the University of Kobe in Japan. So, the PKC was actually discovered in the year 1977. It was originally referred to as the cyclic nucleotide independent proteolytically modified protein kinase from mammalian brain, which was actually named as PKM primarily because magnesium is required for its activation. The full length enzyme was later shown to be activated by calcium and phospholipids and was actually named protein kinase C. C standing for calcium ions, which is fully which is required for activation of this enzyme at very low concentration, and the enzyme was named as protein kinase C primarily to differentiate it from the cyclic nucleotide dependent kinases like protein kinase A and protein kinase G, which activate the cyclic AMP dependent protein kinase and the which refers to cyclic GMP dependent protein kinases. So, like unsaturated diacylglycerol was shown to be an essential activator of protein kinase C, and this particular discovery of linking DAG to the PKC actually signaled a very important role for PKC in the signal transduction pathway, and thus began a very intense investigation of the understanding the role of PKC in signal transduction pathways in leading to the activation of metabolic enzymes, ion channels, as well as transcription factors. The protein kinase C family, ever since its discovery way back in 1977, consists of a number of members, and they are broadly categorized into 3 groups. The calcium dependent PKCs or the conventional PKCs designated as CPKCs. It contains of PKC1, PKC2 and PKC3, also known as CPKC gamma, beta and alpha. There is also calcium independent PKCs, which are actually novel PKCs or NPKCs, and there are also what are known as atypical PKCs or APKCs. And in the next slide I have actually shown the various domain structures of all these various members of the PKC family. Basically they contain what is called as a PKC regulatory domain and a PKC catalytic domain, but we will not go into the details of the various structural details of the PKC family members. There are references which I have listed at the end of this lecture series, and if you read some of these review articles you will get more idea about the structure and function of these various protein kinase C members of this family, but our primary goal here is to understand their role in the regulation of gene expression. So, the protein kinase C is primarily involved in the regulation of diverse cellular processes including growth, differentiation, neural development, synaptic transmission, axonal regeneration, smooth muscle contraction, relaxation, endocrine exocrine secretion, tumor promotion and aging. You can see PKC plays a very important role in regulating a number of cellular processes. But remember, as I keep telling, many of these protein kinases, whether it is protein kinase C or calcium calmodeling kinase or protein kinase C, they all regulate the cellular process through different means, and regulation of gene expression is only one part of it. So, they can directly go and activate certain metabolic enzymes, or they can inhibit metabolic enzymes, they can go and influence certain some structural proteins or cytoskeletal proteins, or they may have an influence on ion channels. But we are not going to discuss any of these aspects. In this lecture series we are going to focus primarily our attention on how these protein kinases involve or involved in the regulation of gene expression, what kind of transcription factors are activated or repressed by these protein kinases, and we will not discuss about their effect on metabolic enzymes, cytoskeletal proteins and ion channel proteins in this lecture series. The importance of protein kinase C in controlling cell division and proliferation was revealed by the action of compounds known as Farbol esters. What are Farbol esters? Actually in the year 1982, the Farbol esters were shown to directly activate protein kinase C, and activation of Farbol esters was shown to lead to translocation of protein kinase C from the cell soluble to the cell particulate form. So, this is a very important discovery in the field of protein kinase C research, and I have shown here the structure of a Farbol esters, and let us see how exactly Farbol esters activate protein kinase C. Now, these tumor promoting Farbol esters such as Farbol or myristate 13 acetate, known as abbreviated as PMA, they actually possess a molecular structure very similar to that of diacylglycerol, and we know diacylglycerol and calcium regulate protein kinase C function. They are the activators of protein kinase C, and these PMAs or Farbol esters have a structure which is similar to the diacylglycerol. Now, these agents can therefore substitute for diag in activating protein kinase C either directly in vitro or in vivo. So, if we treat the cells with these Farbol esters, they directly diffuse across the plasma membrane, directly bind to protein kinase C and activate the protein kinase C signaling pathway. Whereas, the normal effector signaling molecules bind to their cognate receptors present on the cell surface, which in turn lead to the activation of specific G proteins, and G proteins in turn activate phospholipase C, and phospholipase C then produces diacylglycerol, and this diacylglycerol now activates protein kinase C. So, if we treat cells with Farbol esters, it completely bypasses this signaling mechanism, and these Farbol esters directly go and bind to protein kinase C and activate. So, like diacylglycerol, Farbol esters dramatically increase the affinity of protein kinase C for calcium, resulting in its complete activation at physiological calcium concentrations. So, since Farbol esters are very slowly metabolized, they persist in the tissues for much longer than DAG, and therefore cause prolonged activation of PKC, leading to uncontrolled cell proliferation. Now, PKC plays a very important role in the regulation of cell proliferation, and if PKC is continuously activated, it results in uncontrolled cell proliferation. Okay. So, if we have molecules like Farbol esters, or if we treat cells with molecules like Farbol esters, since these Farbol esters go and directly bind to protein kinase C and they are not degraded in the cell very rapidly, it results in continuous activation of protein kinase C, and as a result there is a continuous uncontrolled cell proliferation leading to cancer. So, a very important aspect of protein kinase C research was the discovery that the protein kinase C is actually anchored to membranes by specific proteins, and the individual isozymes are often localized to specific subcellular sites following their activation. For example, in the year 1991, mostly Roshan and his co-workers actually identified PKC anchoring proteins and named them as receptors for activated C kinases, or RACs. So, there is another level of regulation as far as protein kinase C is concerned. Depending upon whether this protein kinase C is anchored to these RACs or not, there could be regulation. So, the protein kinases may remain inaccessible for regulation when they are anchored to this protein, and when they are removed from these anchoring proteins, then they become available to do their catalysis and therefore regulate various cellular processes. So, depending upon the various tissue type, different kinds of RACs actually hold these protein kinase C molecules in a specific region, subcellular locations, and this constitutes another important regulation of protein kinase C signaling. There is a very nice paper that actually discussed about the RACs in detail, the PNAS paper in 1991. So, let us now briefly discuss about how protein kinase C regulates cell proliferation. So far we have discussed how molecules binding to their GPCRs ultimately result in the activation of trimeric G proteins, and these trimeric G proteins, which contain the GQ alpha subunit, now activates phospholipase C, and phospholipase C now cleaves the phosphatidyl inositol bisphosphate, or PAP2, into diacylglycerol and inositol triphosphate. Inositol triphosphate in turn goes and then activates the calcium channels in the endoplasmic reticulum. It results in a huge increase in intracellular calcium release, and this calcium along with the diacylglycerol now goes and binds to protein kinase C and then activates the protein kinase C. Now, once the protein kinase C is activated, what does it do? One of the main functions of protein kinase C, especially the major mechanism by which protein kinase C activates cell proliferation pathways, is actually by activating the expression of very specific set of genes. So, the protein kinase C acts as a regulator of gene expression primarily by phosphorylating certain specific transcription factors, which then activate specific genes which are involved in the cell proliferation pathway, leading to cell division and cell proliferation. So, this is one of the important physiological processes regulated by protein kinase C. Now, it turns out cell proliferation in general happens in two different phases. One is called as the early response, and another is called the delayed response or the late response. Now, very interestingly, the protein kinase C plays a very important role both in the early response as well as in the late response. Now, let us see, this is a very brief schematic sketch that actually tells you how protein kinase C acts as a regulator of cell proliferation. Turns out, the moment protein kinase C is activated by diacylglycerol and calcium, protein kinase C now goes and phosphorylates on specific transcription factors, which turns on the expression of a set of genes known as immediate early genes. Now, these immediate early genes are so named because their expression is induced within minutes after this growth promoting stimulus. Within 15 minutes or 20 minutes after you add a particular stimulus which is known to be involved in cell proliferation, this set of genes are activated. These immediate gene products are often referred to as the third messengers. They in turn again are phosphorylated they get translated, that is when the immediate early genes are transcribed, they are translated to specific proteins. Usually these proteins again are transcription factors which are again phosphorylated by protein kinase C and therefore activated. They in turn go and activate all the genes involved in the cell proliferation. So, activation of immediate early genes as well as activation of genes involved in late response by protein kinase C actually marks the mechanism by which protein kinase C promotes cell proliferation. Let us discuss in detail how exactly the cell proliferation pathways operate. Let us say take an example. I am sure many of you are familiar the way mammalian cells are cultured in vitro, in laboratory, in CO2 incubators. Usually you have Petri plates where you take these mammalian cells like NH3 fibroblasts, or it could be primary cells, or it could be transformed cells, and usually whenever you want to culture these cells or cell lines in laboratory, you do it in presence of specific media which contains the carbon source like glucose and various other ions and so on and so forth, but it also contains a very important molecule called serum. Usually you either have a fetal bovine serum or fetal calf serum, about 5 to 10%, and usually mammalian cells are cultured in presence of this serum. Now, what does serum do? Serum is actually essential for the cells to proliferate. It turns out the serum contains a number of growth factors, and when these growth factors bind to the specific receptors, it results in the activation of specific kinases including the protein kinase C, and this in turn activates the immediate early response gene as well as the late response genes, and that is how the cell proliferation program is initiated. For example, as I mentioned earlier, the early response phase, as soon as protein kinase is activated, it first activates a set of early response phase, which actually involves phosphorylation of proteins called as serum response factor or SRF. Now, SRF is a very important transcription factor which has binding elements in the promoters of immediate early genes such as CFOS, JUNB, FOSB, EGR1 and so on and so forth. So, all these genes known as the immediate early genes in their promoter contain response elements for the serum response factor, and as a result, as soon as the serum response factor is phosphorylated by protein kinase C, the phosphorylated SRF now goes and binds to the serum response elements of these genes and activate the expression of these genes, and as a result all these proteins are actually made. And all these proteins like FOS, JUN and EGRF, they are all transcription factors, and once these transcription factors are made, they are again phosphorylated by protein kinase C and therefore activated, which in turn go and then activate target genes that are involved in various cell proliferation programs. So, this is how protein kinase C primarily activates cell proliferation pathways. So, as I said mentioned earlier, protein kinase C is involved in the phosphorylation of transcription factors of not only the early response phase, but also of the late response phase. That means, it not only activates or phosphorylates transcription factor like serum response factor, but it also acts on proteins like CFOS and NF kappa B, which are actually synthesized following the activation by SRF. So, the immediate early genes are the genes which are activated transiently and rapidly in response to a wide variety of cellular stimuli, growth proliferating stimuli. They represent a standing response mechanism that is activated at the transcription level in the first round of response to stimuli before any new proteins are synthesized. So, you can see the immediate early gene activation actually requires no protein synthesis. There are already proteins like serum response factor in the cytoplasm, and they are phosphorylated by protein kinase C, and that now triggers transcription of immediate early genes. So, transactivation of immediate early genes does not require any protein synthesis. It basically requires phosphorylation of pre-existing transcription factors like SRF. So, these immediate early genes are distinct from late response genes, which can only be activated later following the synthesis of early response gene products. Like when the serum response factor now activates the expression of genes like CFOS, EGR1 and CJUN, they in turn activate the target genes which are actually involved in the cell proliferation. So, the second strategy that are activated by the immediate early gene products are known as the late response genes. So, the immediate early genes have been called therefore the gateway to genomic response, because without that cell proliferation is not possible. So, many of the immediate early gene products are therefore naturally transcription factors or DNA binding proteins. So, one of the major functions of protein kinase C is to phosphorylate transcription factors like SRF, which is a transcription factor and therefore goes and binds to the promoters of immediate early genes and activate their expression. And these immediate early gene products are also transcription factors, because they have to now go and actually regulate the various target genes which are directly involved in promoting cell proliferation. Some of the best characterized immediate early genes are CFOS, CMUC and CJUN, and they are all transcription factors. And protein kinase C plays a key role in the activation of all these immediate early genes. I have now given a very brief schematic of how actually protein kinase C activates whatever I have discussed so far. Like I mentioned here, once the protein kinase C is activated in the cell by enzymes like phospholipase C and through the binding of diacylglycerol and calcium, protein kinase C now phosphorylates serum response factor, and the phosphorylated serum response factor now goes and binds to the serum response elements present in the promoters of immediate early genes, which could be CFOS, NF kappa B, EGR1 and so on and so forth. And as a result of this, all these immediate early genes are transcribed, they are translated, and the proteins like CFOS, CJUN and NF kappa B are again phosphorylated by protein kinase C and therefore are activated. And for example, in the case of NF kappa B, the protein kinase C actually phosphorylates an inhibitor of the kappa B called the I kappa B, and therefore the I kappa B can no longer interact with NF kappa B. Therefore, NF kappa B now becomes active, goes to the nucleus and activates the target genes involved in cell proliferation. Whereas in the case of the JUN and FOS, both these proteins are directly phosphorylated protein kinase C, and in the phosphorylation actually takes place in the transcription activation domain, and as a result they go and bind to the AP1 response elements of the group genes involved in cell proliferation, leading to their activation and leading to cell proliferation and cell division. Now, among these proteins, the C-Phos, C-Jun are called as the AP1. We have already studied in detail when we studied about the transcription factors, their various DNA binding domains, transcription activation domain in our earlier lectures. If you remember very well, the AP1 or C-Phos or C-Jun are the transcription factors which contain the basic region and leucine zipper through which they go and bind to a sequence called TGA CTCA. And I also mentioned in my lecture on the cyclic AMP response, this sequence or the AP1 response element TGA CTCA is very slightly different from the cyclic AMP response element or the Krebs binding site, which is TGA CGTCA, whereas AP1 binds to TGA CTCA. G is missing. Now, the other very important immediate early response gene is a gene that codes for a transcription track called EGR1. It is so named because it actually means early growth response gene product or EGR1. It is also known as ZIF268, NGF1A, CROX24 or TIS8. It is basically a zinc finger transcription factor and has been shown to be immediately induced when cells are exposed to stimuli that promotes growth and cell differentiation. EGR1 is induced very rapidly within minutes of a stimulant and is also rapidly decayed within hours. So, many of these immediate response gene products, their levels are very finely controlled inside the cells, and if these levels are tampered with or if they are overexpressed, it can actually lead to cancer. So, the levels as well as the activity of the early growth response genes has to be regulated in a very fine manner in the normal cells, and the one thing that happens in the cancer cells is that many of these transcription factors are overexpressed, and as a result they continuously promote cell proliferation. Now, I have given just one example actually taken from this particular article just to give you an idea how the protein kinase C signaling is linked to the activation of EGR1 expression. For example, in the case of arteries and veins, blood vessels, the EGR1 signaling has been shown to play a very important role in vascular stress. We like acute stress like hypoxia, ischemia, as well as chronic stress like atherosclerosis and restenosis. All these stress signals activate the PKC beta, one of the members of the PKC family of kinases, and the PKC beta activation results in the activation of EGR1, which then results in the synthesis of various cytokines, chemokines, procoagulant molecules, addition molecules and so on and so forth, leading to a number of pathological processes like inflammation, migration as well as proliferation, leading to vascular dysfunction and tissue injury. So, activation of the PKC signaling pathway through the EGR1 pathway plays a very important role in vascular stress and managing a number of vascular diseases. Now, let us try to spend some time to understand what is the role of PKC in the regulation of various T cell functions. So far I gave you one of the important pathways which are activated by PKC is the cell proliferation pathways involving immediate early response genes, which like EGR and C-Phos, which in turn go and activate the actual target genes involved in cell proliferation. Now, let us try to understand another example of how protein kinase C is actually involved in the regulation of various T cell functions. Now, here is a cartoon that actually I have drawn to basically tell you how the protein kinase C plays a very important role in the synthesis of cytokines like interleukin 2 following the binding of antigen presenting cell to the T cell receptor. The APCs containing the antigen bound to the MHC molecule, when it docks with the T cell, it results in the activation of the T cells and results in the production of cytokines like IL-2, and this cartoon basically shows how protein kinase C plays a very important role in the activation of IL-2 gene expression when the antigen presenting cell docks with the T cell receptor. The docking of the antigen presenting cell with the T cell receptor activates a number of signaling pathways. It is called the T cell receptor signaling pathways. We will not go into all the details because in order to understand all the T cell signaling, we also need to understand other signaling mechanisms which we are going to discuss in the end. Suffice to know at this stage that one of the signaling pathways that are activated by the docking of the antigen presenting cell or binding of the antigen presenting cell MHC molecule with the T cell receptor is the activation of, leads to the activation of protein kinase C, and this protein kinase C through the MAP kinase pathway now phosphorylates the inhibitor of kappa B or the I kappa B, and once the inhibitor of I kappa B is phosphorylated, it is ubiquitinated and is degraded, and therefore the NF kappa B, which is actually held in inactive form in the cytoplasm, now becomes activated, and that NF kappa B now goes and binds to the promoter of IL-2 gene and then activates its expression. Now, for the efficient activation of IL-2 gene, the IL-2 gene has to be activated not only by NF kappa B, but also by other transcription factor like NF-AT, AP1 and so on and so forth. So, the promoter of IL-2 gene contains binding sites for all these 3 transcription factors like AP1, NF-AT and NF kappa B. And one of the mechanisms by which NF kappa B is activated is through the protein kinase C signaling pathways. But the NF kappa B is a very important, very complicated transcription factor which is activated by multiple signaling pathways. The same way, similarly, for example, for the efficient activation of the IL-2 gene, not only you need NF kappa B, you also require other transcription factors NF2 and AP1, which are activated by other pathways. Now, the AP1 is activated by the RAS MAP kinase pathway, but the AP1 also is directly phosphorylated by protein kinase C, and therefore it can be activated directly. Whereas the NF-AT is actually activated by a calcium dependent pathway, which I will discuss in the next slide, and which results in the activation of calcineurin, which is a phosphatase which actually dephosphorylates NF-AT, and NF-AT is inactive in the phosphorylated form, is active in the dephosphorylated form, and one NF-AT is dephosphorylated by calcineurin. NF-AT now goes into the nucleus, binds to its response element and activates transcription. So, 3 important transcription factors which are required for IL-2 gene expression, namely the NF kappa B, NF-AT and AP1 are activated by multiple signaling pathways during the T cell receptor activation by the antigen presenting cell, and of which the NF kappa B activation directly involves PKC mediated MAP kinase pathway activation, and PKC can also directly phosphorylate AP1, whereas NF-AT is activated rather indirectly, whereas the calcium, which is actually released by the action of phospholipase C, the calcium that is released by the inositol triphosphate, which is produced by the phospholipase C activation, the calcium then activates the calcineurin. The calcineurin in turn dephosphorylates NF-AT. NF-AT then goes and binds, go to the nucleus, binds and activates the transcription of the gene. Now, I want to just spend one important maybe 1 minute on this calcineurin pathway, because it has very important role in certain biomedical applications. This is again a cartoon which actually a very simplified version of what I have just told you. During the T cell receptor signaling, the phosphatidylinositol bisphosphate is cleaved by phospholipase C into inositol triphosphate and diacylglycerol. Diacylglycerol now activates protein kinase C. Protein kinase C through the MAP kinase signaling now activates AP1 as well as NF kappa B. Now, the inositol triphosphate that is also produced by the phospholipase C action cleavage of phosphatidylinositol bisphosphate now releases the intracellular calcium levels. This calcium now binds to calmodulin, and the calcium calmodulin activate calcineurin, which is actually a phosphatase. Calcineurin now dephosphorylates NF-AT, and the dephosphorylated form NF-AT now goes into the nucleus, binds to the response element together with AP1 as well as NF kappa B, now activates, induces IL-2 gene expression. Now, a very important aspect about this, I want to spend some time to just to emphasize the fact that understanding these signaling pathways and understanding the various transcription factors which are activated or repressed by the activation of the signaling pathways is very important for you to develop novel drug molecules, because many of these components of the signal transduction pathways, either at the transcription factor level or at the kinase or phosphatase level, they are all very important drug targets. Here is one example. Now, calcineurin, as I told you, is very essential for the activation of NF-AT, and the very important immunosuppressive drugs like cyclosporine actually act as inhibitors of calcineurin. And as you can see in the next slide, cyclosporine is a very important immunosuppressive. Those people who undergo something like kidney transplantation or organ transplantations, for example, they usually are treated with immunosuppressants because you want to prevent rejection of the transplanted organs. So, one of the molecules which are actually given for drugs which are given to these patients are immunosuppressants like cyclosporine, and cyclosporine primarily acts by inhibiting the calcineurin. Therefore, NF-AT is not activated, therefore IL-2 is not synchronized, and therefore the antigens or the alloantigens which are present in the tissues are not recognized by the immune system, and therefore there is immunosuppression. So, a major consequence is the inhibition of the activation of transcription factor NF-AT, thus preventing the rejection of foreign organs or bone marrow or even kidney transplantation by disrupting the signaling pathway that activates T cell. So, you can see how understanding the signal transcription pathways and how understanding which transcription factors are activated by which signaling pathway is very important for you to develop drug targets and novel drug molecules. And by knowing that activation of the PKC signaling pathway results in the increase in calcium levels. This calcium levels in turn through the calcium calmodulin pathway results in the activation of calcineurin, and calcineurin now dephosphorylates NF80 and therefore activating NF80, and this is very essential for IL-2 expression. And this knowledge has now led to the development of immunosuppressant molecule like cyclosporin, which actually inhibit calcineurin, and therefore the IL-2 expressions can be prevented. So, understanding these signaling pathways and how transcription factors are activated by these signaling pathways is very essential for developing new drug molecules as well as for the identification of drug targets, and this is a billion-dollar industry. So, in order for you to understand and appreciate gene regulation, you have to understand signaling molecules or signaling pathways. If you do not understand how cells communicate with extracellular signaling molecules and how extracellular molecules through a various cascade or signaling pathways affect specific gene expression programs, without this knowledge we cannot appreciate how gene expression takes place and how transcription factors or transcription or gene expression programs can be made use of for the benefit of mankind. The protein kinase, so what I have told you so far is that protein kinase C plays a very important role in the antigen presentation, leading to the activation of the T cell receptor following antigen presentation, and cytokines like IL-2 are actually synthesized primarily by activation of specific signaling pathways involving protein kinase C. Now, a number of evidence actually indicate that PKC plays a very important role in the not only in the activation of T cell receptor but also a number of T cell signaling pathways. And one best way of actually demonstrating these to see what kind of protein kinase is actually expressed in T cells, and then you knock out this particular PKC and see what is the effect of this knockout of PKC on the T cell receptor function. In fact, such a thing has been done. For example, one of the PKC isoforms which are actually expressed in high levels in primary T lymphocytes and muscle is called PKC theta, which is a member of the novel PKCs. I told you in the beginning there are 3 PKC subfamilies, and PKC theta is a novel PKC, member of a novel PKC subfamily of genes. And when you actually knock out this PKC theta, which is a major isoform of PKC which is expressed in T cells, in these knockout T cells, the naive T cells display defect in T cell activation due to the lack of expression of NF80, NF-kappa and AP1, clearly indicating that protein kinase C plays a very important role in the activation of all these 3 transcription factors, and therefore if you knock out PKC, you will not see the activation of these transcription factors, and therefore you do not see IL-2 expression. And transcription factor NF-kappa B and AP1 are the primary physiological targets of PKC theta. I have already mentioned in the previous cartoons how NF-kappa B is activated by PKC pathway. NF-kappa B is activated primarily by the phosphorylation of the I-kappa B by the MAP kinase pathway involving protein kinase C, and when I-kappa B is phosphorylated and degraded by ubiquitin, NF-kappa B now goes in the nucleus and binds to the NF-kappa B response elements of the IL-2 gene. Similarly, AP1 is a direct target of protein kinase C, or it can also be activated through the MAP kinase cascade. So, NF-kappa and AP1 are direct targets for protein kinase C, and efficient activation of this transcription factor by PKC theta is required for the integration of T cell receptor and CD28 co-stimulatory signals. I'm presenting a very simplified version of activation of T cell receptor signaling involving PKC signaling. It's much more complicated. It involves convergence of multiple signaling pathways, but the idea is not to confuse the listener, but to actually understand at least one aspect of T cell receptor signaling. And what am I trying to convey? The message to you is that one aspect of T cell receptor signaling involves protein kinase C, and protein kinase C is essential for the expression of IL-2. This is interleukin 2 expression, and for interleukin 2 expression you require transcription factors like NF-kappa B, AP1 and NF80, and at least 2 of these transcription factors are activated by the PKC pathway. And PKC theta cooperates with protein serine threonine phosphatase calcineurin in transducing signal leading to the activation of JNK, NF80 and as well as the IL-2 gene. So, protein kinase C plays a very important role in the T cell functions. In fact, a number of studies actually shown that PKC theta plays a very important role not only in T cell activation but also in T cell survival, apoptosis as well as IL-2 production. So, as far as T cell activation is, so using this kind of a PKC theta knockout mice, it has been very well shown that the protein kinase, the PKC theta plays a very important role in T cell activation because it is essential for the activation of transcription factors like NF-kappa B, AP1 and NF80. It is also plays a very important role in T cell survival. For example, it is involved in the activation of BCL-XL and BCL2 genes, which are involved in the apoptosis as a T cell survival, as well as in the apoptosis it regulates the expression of FOS and FOS ligand and it is also involved in T cell proliferation, as I just told you how IL-2 expression is directly regulated by the influence of transcription factors. So, protein kinase C plays a very important role in a number of T cell functions. What I would like now mention is to just take you a few minutes to tell you that there are umpteen number of examples to actually demonstrate how PKC plays a very important role and what kind of signaling pathways are activated by signaling by protein kinase C, ultimately leading to the activation of specific transcription factors and specific target genes. And there are actually pathways which have been worked out, and these pathways are actually present in a number of journal websites, like for example some of the signaling pathways which are activated by protein kinase C are what are called the myocyte adrenergic pathway, granule cell survival pathway, differentiation pathway involving PC12 cells. So, I am just going to show some of these cartoons taken from the journal websites to just tell you how PKC plays a very important role in these various signaling pathways. For example, PC12 is actually a pheochromocytoma cell line, which is a clonal cell line derived from a neuronal cell line, and this is derived from the transplantable rat adrenal pheochromocytoma cells, which responds reversibly to nerve growth factor. So, if we take PC12 cells and treat them with nerve growth factor, they differentiate into neurons. Okay. Now, using this model system, people have actually demonstrated what are the signaling pathways involved in this differentiation program. How do these cells differentiate into neurons? And it turns out the protein kinase C plays a very important role in this nerve growth factor mediated differentiation of PC12 cells into neuronal cells. In addition to the nerve growth factor, another important effector molecule called pituitary adenylate cyclase activating polypeptide PACAP is also an adrenomedullary neurotransmitter, which is also has been shown to cause PC12 differentiation. Now, this mechanism by which both this NGF as well as PACAP activate this program, differentiation program of PC12 has been very well worked out and has been now summarized in this particular cartoon taken from the Science Signaling website, and you can see the nerve growth factor and the PACAP shown in the blue arrows here activate two distinct signal transduction pathways which converge, and all like the NGF pathway actually involves the activation of the protein kinase C here, which through again the MAP kinase pathway activates ultimately the transcription like C-Jun, EGR1 and so on and so forth, or AP1, and both these factors through two distinct signaling pathways, one involving the G protein coupled signaling pathway as well as the CREB pathway can activate the differentiation program, and the nerve growth factor primarily activates through the PKC pathway. So, using these kinds of model systems, people have actually drawn the signaling networks, and such signal transduction pathways have been worked out for a number of physiological processes, and I am just going to show two more examples to actually show the PKC involvement in such signaling pathways. So, PKC plays a very important role in the differentiation of these PC12s into neurons. Again, the angiotensin 2 stimulated signaling through G proteins and beta arrestins again involves the PKC pathway. I don't expect you to go through all the details here, but just you can see when these molecules bind to the receptors, it results in the activation of phospholipase C, results in synthesis of inositol triphosphate and diacylglycerol, and inositol triphosphate results in the calcium release, and both DAG and calcium now activate PKC, leading to the activation of downstream signaling molecules, ultimately leading to the activation of the early growth response. This is just the details of what exactly is angiotensin 1 receptors. I will not go into the details. Suffice to know that stimulation of the receptor with the peptide ligand angiotensin 2 result in the activation of the GAQ11 subunit of G protein, trimeric G proteins, leading to the activation of the protein kinase C. This leads to the activation of ERK, or the extracellular receptor kinases cascade. This extracellular receptor kinase then translocates nucleus and stimulates the transcription pathways governed by the ELK activity and the EGR1 transcription factor, which is the early growth response gene product. Similarly, insulin-like growth factor, again a very important molecule involved in cell proliferation, again acts through a PI3 kinase pathway. This is the receptor tyrosine kinase pathway. We are going to discuss in the next class what are these receptor tyrosine kinases. So, again when the insulin growth factor binds to its receptor, it results in the activation of a receptor tyrosine kinase, and through the PI3K signaling pathway again PKC is activated. So, PKC is not only activated by the trimeric G proteins, PKC can also be activated by the receptor tyrosine kinase pathways, and the activation of protein kinase again results in the activation of MAP kinases. The 3 MAP kinases are shown here, the MAP3 kinase, MAP kinase and MAP kinase, ultimately leading to the phosphorylation of transcription factors and activating specific gene expression programs. So it is not possible to discuss all the signal transduction pathways, so I have just given a few examples to just tell you how PKC plays a very important role in activating specific gene expression programs. Now PKC is of extraordinary biomedical importance because a number of disease processes are attributed to the improper activation or improper regulation of protein kinase C. For example, PKC has been shown to be involved in insulin resistance and diabetes. For example, in obese individuals, fatty acids and acetyl-CoAs, ceramides, they increase, and the diacylglycerol, as a result, all these molecules like fatty acids, acetyl-CoAs and ceramides, they activate the PKC signaling pathway through the diacylglycerol activation. As a result, the serine phosphorylation of the IRS, which is a very important signaling molecule in the insulin receptor signaling pathway, the result to impaired insulin signaling, leading to diseases like diabetes. So PKC plays a very important role in insulin resistance. Similarly, PKC plays a very important role in diabetes. Hyperglycemic control in diabetes is a key to preventing development and progression of vascular complications like retinopathy, nephropathy and neuropathy. Advanced stages of diabetes patients, one of the major complications in diabetes patients is retinopathy, nephropathy and neuropathy. These are all very complicated manifestations of diabetes, and it has been shown in all these ailments increased activation of the diacylglycerol protein kinase C pathway has been identified as a major cause for the vascular tissues damage from the diabetic animals and those vascular cells which are exposed to elevated glucose levels. So when you have high levels of glucose in your blood, it results in the activation of the diacylglycerol protein kinase C pathway, resulting in all these kinds of diseases like retinopathy, nephropathy and neuropathy. Vascular abnormalities associated with glucose-induced PKC activation leading to increase in this of DAG includes altered vascular blood flow, extracellular matrix deposition, basement membrane thickening, increased permeability, neovascularization. These are all various clinical manifestations of advanced stages of diabetes. So preferential activation of PKC beta isoform by elevated glucose is reported to occur in a variety of vascular tissues of diabetes patients. So what is the significance of this knowledge? People are actually trying to see whether these kinds of patients can be treated with some of the inhibitors of the protein kinases. For example, a molecule like LY333531, a potent inhibitor of PKC beta isoform, has been shown to have a potential to be an orally effective non-toxic therapy for producing significant improvements in diabetic nephropathy, neuropathy and naturopathy as well as cardiac dysfunction. So inhibitors of protein kinase C have tremendous potential in treatment of diseases like diabetes, especially some of the problems involved in diabetes. Similarly, gonadotropin hormones, FSH, LH, they all activate through these G protein coupled receptors, the 7 transmembrane proteins. Again, the GQ dependent protein kinase C activation plays a major role in the GnRH stimulated extracellular receptor kinase pathway of making physiological responses. I will not go into the details, just to tell you that the gonadotropin action also involves the PKC signaling pathway. Similarly, the most common PKC isoforms deregulated in cancer are alpha, beta and gamma. These are the classical PKCs, and all these when these PKCs are misregulated or dysregulated, it results in cancer. For example, the two main effects of deregulation of PKC in cancer are on mitogenic signal and apoptotic signals. The mitogenic signals, for example, aggressive breast cancer cell lines have elevated levels of PKC epsilon levels that make them more proliferative, invasive and motile. Similarly, PKC alpha has been linked to decreased proliferation in gastric cancer and increased proliferation in gliomas. PKC delta is usually associated with growth arrest and tumor suppression. It can also promote survival and in some cases metastatic potential such as melanoma, breast and lung cancer cells. So PKC deregulation plays a very important role in the activation of number of mitogenic signal. Similarly, PKC epsilon is known to suppress apoptosis in prostate cancer cells and promote the survival through inhibition of mitochondria-induced caspase activation in lung cancer. Similarly, inhibition of PKC alpha induced apoptosis in glioma cells. Similarly, PKC delta plays a very important role in apoptotic mechanism induced by a number of anti-cancer drugs. So you can see understanding the pathways by which protein kinase is activated can actually lead to development of inhibitors of this PKC and see whether you can actually develop some novel anti-cancer compounds. Just as the phosphorylation esters are very potent activators of protein kinase C, people have also come up with a number of analogues of diacylglycerol, and one such analog is called bryostatin 1, which is also a structural analog of diacylglycerol, and bryostatin 1 has also been shown to have anti-tumor activity on certain kind of leukemic cells. And bryostatin 1 is a very potent activator of PKC. The structure is shown here, taken from this particular paper. And bryostatin 1 not only mimics some of the 4-ball ester responses and antagonize those actions of 4-ball esters that it cannot produce. So variants of diacylglycerol by structural analogs of diacylglycerol like 4-ball esters and bryostatin are now being used actually to promote the anti-tumor activity of protein kinase C, and in certain tumor cells when you activate protein kinase C using these molecules, it can lead to apoptosis or cessation of cell proliferation. So what I have listed in the next few slides is some of the original research articles. Especially in this slide, I have listed the very important key research paper that actually led to the discovery of protein kinase C, especially by the Nishizuka group who are actually credited with the discovery of protein kinase C. These are the first 2 JBC papers which actually led to the discovery of protein kinase C way back in 1977. The activation of PKC by phosphoresters was first shown in these 2 JBC papers, again by Nishizuka group in 1982. It's a very excellent paper in Nature which actually shows how PKC theta is required for the T cell receptor induced NF-kappa B activation in mature but not immature T lymphocytes. The paper clearly demonstrates that PKC delta is essential for T cell receptor mediated T cell activation, but is dispensable during the T cell receptor thymocyte development, and T cell receptor initiated NF-kappa B activation was absent in PKC theta knockout mature lymphocytes but was intact in thymocytes. So using PKC theta knockout mice, these authors have clearly demonstrated what is the role of PKC in T cell signaling and T cell function. There is again a very interesting paper which actually tells for what is called as a PKC syndrome, how by improper diet you can actually develop into what is called as a PKC syndrome, and the author very nicely says that it can be anticipated that various aspects of modern diet and lifestyle such as high fat diet, low omega-3 intake, abdominal obesity, enhanced adipocyte liposis, secondary to insulin resistance, diabetic hyperglycemia will tend to increase diacylglycerol production and thus enhance the activity of PKC. So if you now start eating high fat diet, this high fat diet actually produces a lot of diacylglycerol, and this diacylglycerol can activate protein kinase C, leading to all kinds of problems. So depending upon what diet you take can also influence whether protein kinase C pathway can be activated or not. Two very important reviews to celebrate 25 years of discovery of protein kinase C. Is a very nice review article in Pharmacological Research. Happy birthday protein kinase C. A nice review of all the protein kinase C research that went on, and a very more recent article in 2010, protein kinase C positive signal, actually gives the overview of the importance of various PKC signaling pathways. Again, the role of protein kinase C in the early growth response pathway, how it can lead to ischemia, atherosclerosis and restenosis is discussed in this particular article. So what I have covered in the last 3 lectures is some of the aspects of the G protein signaling pathways. I tried to cover the GS and the GI pathway, but due to the paucity of time I am not going to cover the remaining pathways, but I suggest you read this excellent review in Science which appeared in 2003, in which he very briefly discussed about the various G protein trimeric G protein signaling pathways and how these G protein signaling pathways can result in the regulation of number of physiological processes. I think I will stop here.